Hello guys and welcome back to Excelsior Academy. So today we are going to do the last topic of the cell signaling. Okay. So there are other signaling also like between the like for example the host parasite signaling and also that will be taken differently. This is within the cell, like inside the cell how the signal is being passed. Okay. So for this now the last topic is like where I was discussing about like how the crosstalk or the network of the signaling is happening. So as such in the reference book is not given much, but if you see, if you get a chance to read any research paper, I am not sure how much you will understand though, but the gist of the story is that like all the signaling pathways, okay, it is not that only one at a time one pathway is getting activated. It is the entire pathways. अगर suppose 100 pathways हैं signaling के, तो वो सारे के सारे activated हो रहते हैं, ठीक है? Because they are in continuous in response with the surroundings in with the environment, right? So depending on that the cell is always functioning like kya karna hai, like to stay in the same stage or not, or should I move? So if I'm moving, so what all proteins are required? So all that coordination is required. Okay, so we'll just see a gist of it. The signaling dynamics and the network. We have so far discussed signaling in terms of linear pathways, which transmit information from the environment to the intracellular targets. Okay, so every time we were saying that the growth factor or any hormone or ligands will come, it will bind to the receptor and the downstream signaling will be taken care of. However, within the cell, it is far more complicated. First, the activities of individual pathways, they are regulated by feedback loops that control the extent and the duration of signaling activity. Now, this is also important, guys, to understand like the extent to which the signal activity is going on. See, it's like if suppose any growth factor is coming and binding to its receptor, we want it to signal, pass the signal and then stop, right? There is a duration. Otherwise क्या होगा कि ये same growth factor पे continuous gene expression करता रहेगा, even though it is not required, right? So we want that the dissociation between the growth factor and the receptor should take place after the signal has been passed, or you know the degradation of the growth factor or the receptor by which the signal is being stopped. ठीक है, तो या फिर डिग्रेडेशन होगा या इट विल गेट phosphorylated so that it loses its affinity to the receptor and stops the signaling. Okay, now in addition, signaling pathways do not operate in isolation. Rather, there is a frequent crosstalk between the different pathways, so that intercellular signal transaction ultimately needs to be understood as an integrated network of connected pathways. Okay. Now look it over here. So this is taken from one of the research paper where they're trying to make all the connections between the pathways. Okay, let's see this example. So here you can see few things, even like a few of the pathways are there which even I'm not sure what it is about. So we have seen about the GSK, TCF pathway, we have seen Wnt pathway, we have seen then ubiquitination is happening, correct? Then we also talked about the FOXO. So as you can see over here, the connections are made between each pathway. And you can see each molecule is interconnected. For example, if it is TCF, it is also connected to the Wnt. It is also connected to this particular ubiquitination. Okay. Same way, the other FOXO. You can see here all different pathways are being again connected. So either it is getting prevented or it is getting activated. So guys, are you getting the story here, like how it is well connected, the crosstalk which we are talking about here? So nothing will come for the exam from here, but it is only for the understanding, like that one single pathway is not taking place. It is all the pathways which are connected to each other and it is happening at the same time inside a cell. Is this much is clear? Of course, this is not going to be, asked in the exam or anywhere because this is just taken from one of the research paper. Now coming to the feedback loops and the signaling dynamics. So the activity of signaling pathway is controlled by the feedback loops which are similar in principle to feedback regulation of metabolic pathways. For example, let's say if threonine is there, from threonine, isoleucine is forming, right? So what is the first step then? It is catalyzed from threonine with the help of the deaminase enzyme. It is becoming alpha ketobutyrate, right? Now, if suppose you have got so much amount of so much concentration of isoleucine present, so what is happening? This isoleucine will, as the end product of the pathway, will go and bind to the threonine deaminase, saying that to inhibit the pathway. Did you get it, guys? This one, Akshaya? This is a feedback inhibition where the end product, when it reaches the particular concentration, it is going to inhibit the enzyme itself. So more the concentration it goes and binds to the enzyme and inhibits the activity of the enzyme, so that the further production or further synthesis of isoleucine is not happening. Clear, guys? Akshaya?
Yes, ma'am.
Yeah. Okay. Now, another example, the feedback inhibition of NF-kappa B, then how this is being done. So this is another example of a negative feedback loop which is provided by the NF-kappa B pathway. So as we know, I-kappa B kinase is there. So it goes in phosphorylates to I-kappa B. So what happens? Then this particular I-kappa B goes for the degradation. Correct? There's a conformational change. Then it is targeted for the ubiquitination and then degraded. Now NF-kappa B is free. It enters your nucleus and the transcription happens. So this is what we studied in the NF-kappa B pathway. Correct. Now, if the feedback inhibition, when we are talking about it, so one of the genes which is coding for the NF-kappa B is also the I-kappa B. क्यों? क्योंकि आपने तो यहाँ पे degrade कर दिया है, but eventually I need this one also. Eventually I need I-kappa B again, right? So this NF-kappa B, when it enters, it is going to transcribe the genes which is required or which is coding for your I-kappa B. Okay, now once again, when the concentration increases, then what happens? It is going to then inhibit your NF-kappa B. Okay, clear? This is negative feedback. यानी कि क्या, जो चीज मैं synthesize कर रही हूँ वही चीज आके मुझे block कर रहा है. This is negative feedback loop. Did you get it? So we know that I-kappa B blocks NF-kappa B. Okay, and NF-kappa B itself synthesizes I-kappa B. Sorry, it acts as a transcription factor for the synthesis of your I-kappa B. So then I-kappa B is formed and then it is translocated into the cytoplasm where it is inhibiting the activity of NF-kappa B. Okay. So is it clear, the feedback inhibition of NF-kappa B, Trishita?
Yes, ma'am. I have one question.
Yeah.
Ma'am, this one kappa B kinase, that is the one which is going to phosphorylate IKB, right? In one kappa B. So how is this kappa B, which is being transcribed, that is also inhibiting this NF kappa B, right? In the step. So my question is it the phosphorylated form that inhibits NF kappa B, or is it I-kappa B which inhibits it?
Acha, okay. I'll say once again. See what happens. Normally, I-kappa B inhibits NF-kappa B. First thing, okay? I-kappa B inhibits your NF-kappa B. Okay. Now, when I-kappa B gets phosphorylated, there's a conformational change, and this is the phosphorylated I-kappa B is meant for degradation then. Now it is clear?
Yes.
Huh?
So then my question is, when it transcribe I-kappa, that is I-kappa B. Now this I-kappa B also is going to inhibit that, right?
I-kappa B, then I-kappa B is then going to inhibit NF-kappa B.
Okay.
Then it will present in the cytoplasm only. It will not allow the entry of NF-kappa B to the nucleus.
Okay.
Huh? Now, I-kappa B kinase is adding the phosphate group to the I-kappa B so that this gets removed. Okay, so this is degraded. Now NF-kappa B is free to enter the nucleus, but NF-kappa B itself is encoding or helping in the synthesis of I-kappa B, which when translocates outside in the cytoplasm goes and binds to the NF-kappa B to stop the transport of NF-kappa B into the nucleus. And that's why it is called negative feedback. Feedback क्या हुआ? पीछे वापस जाना. Negative क्यों? Because this itself is synthesizing for I-kappa B and I-kappa B इसको ही को बंद कर रहा है, prevent कर रहा है. Okay?
Yes, yes.
Now, NF-kappa B is activated by signals that lead to proteolysis of the inhibitor I-kappa B, allowing NF-kappa B to translocate to the nucleus and induce expression of its target genes. Now, अभी देखो हमने लास्ट टाइम पढ़ा था NF-kappa B signaling and TNF का signaling. Now we are going to see together. So यहाँ पे क्या हुआ? So these two signaling pathways are merging together. So your tumor necrosis factor is coming, binding to the tumor necrosis factor receptor. It is activating the adaptive proteins. Now these adaptive proteins are activating your I-kappa B kinase, which phosphorylates I-kappa B. So I-kappa B goes for the degradation, right? And NF-kappa B is then recruited into the nucleus for the transcription of the genes. Okay? Clear? So what is happening? Question आना चाहिए I-kappa B kinase कहाँ से आ रहा है? So that is because of the TNF. Clear now? So हमने पहले ये अलग पढ़ा, now together.
And ma'am, what is the function of adapter there?
So these adapter proteins, could be anything. So before we talked about SH2 domains, SRC proteins, do you remember?
Yes, ma'am.
Yeah, so it could be any adapter protein. So that's why they have written as adapter, because it depends on what the tumor necrosis factor is bound to which receptor of TNF. Accordingly, these adapter proteins will get activated. So based on that, they have just mentioned common, generic name adapter proteins, which is activating your I-kappa B kinase. Okay.
Yes, ma'am.
So one of the target genes induced by NF-kappa B encodes I-kappa B. So NF-kappa B signaling leads to the synthesis of new I-kappa B, which inhibits continued NF-kappa B activity. Okay, so this regulation is critical because the extent and duration of NF-kappa B activity can determine the transcriptional response of the cell. So see, it depends again, कितना time लगेगा I-kappa B को synthesize होके cytoplasm में आके NF-kappa B activity को stop करने का. ठीक है? So that is all determined, like how NF-kappa B signaling is strong or weak, how much it is amplified. So depending on that, it is going to have its the duration of the activity. Okay, so we know that, see, inside the cell everything is happening very fast, but it depends on how much it is amplified. So inside the cell, when you say amplified, means what? It is the number of NF-kappa B transcription factors which got translocated inside the nucleus. ठीक है? तो जैसे कि for example, in the case of GPCR, when we were talking about, in the case of the GPCR, so that time we said, right, eventually resulting in the increased concentration of CAMP. ये कितना ज़्यादा है, acting as a secondary messenger. So it is going to activate, let us say that from one growth factor of one signal, one GPCR was activated. It activated at around 10 adenylyl cyclase. 10 adenylyl cyclase से हमें कम से कम 100 CMP मिला, and now this is going to activate your 1000 PKA, protein kinase A. So those many catalytic subunits will enter, and we can get that many the transcription factors getting activated and the expression of the genes. So this is all about the amplification. So more the amplification, it means more the duration of the signal. Obviously, simple thing, right? जितना ज़्यादा amplified होगा, उतना ज़्यादा duration होगा उस particular signal का. Okay, now signaling by so this you don't have to remember. Okay, I'll tell you like concept point important hai for the exam also. With respect to that also I'll mention. So, but otherwise this is just for your understanding, like how the cell signaling is happening inside the cell together with the other pathways also. So signaling by the ARK MAP kinase provides another example of the importance of the duration of signal. So in a well-studied model of cell differentiation in response to nerve growth factor, ERK signal can lead either to cell proliferation or to neuronal differentiation depending on the duration of ERK activity. Now let's see. Agar ERK activity is for 30 to 60 minutes, then it is for cell proliferation. But if it is for 2 to 3 hours, then it is going for your neuronal differentiation. Okay, so it is again, it is the duration, how much it is amplified, it is going to determine the fate of the cell. Although the mechanism by which these differences in the duration of ERK activity lead to such distinct biological outcomes remains to be understood, it is clear that quantitative considerations of signaling activity are critical to cell response. So apart from this, like amplification, the duration, see, we also need to understand that when these longer duration is happening, it means what the other proteins are also getting involved, whether phosphorylated or acetylated, depending on the requirement. It is not that the same proteins are involved in both the cases. There might be additional proteins involved. Okay. What I want to say is that if suppose we are talking about the ERK pathway, let's say for the cell proliferation, if I'm saying that total downstream 5 proteins are involved, in the same ERK pathway, which is meant for the neural differentiation, which is happening for 2 to 3 hours, what I want to say is that 15 proteins are more involved. Okay. Because that differentiation should come in among the pathways output. कि अगर सेल प्रोलिफरेशन हो रहा है तो ठीक है, सेल आपका डिवाइड होना चाहिए. अगर वो डिफरेंशिएट हो रहा है तो देयर आर अदर पर्टिकुलर फंक्शंस आल्सो, लाइक इट नीड्स टू डिफरेंशिएट एंड गेट डिफरेंशिएट होना मतलब क्या, हैविंग स्पेसिफिक फंक्शन, राइट? सो अगेन so many those particular proteins will be getting introduced or gene expression is happening, and that's why many other proteins are also involved during the cell signaling. Did you get this point which I wanted to explain? Yes, Tanvi, clear hai kya? Should I repeat this point?
Yes, ma'am.
I mean, to repeat, or is it clear?
You can repeat. Yeah.
Acha. Okay. So ERK for cell proliferation is taking 30 to 60 minutes. So इसका मतलब यहाँ पे जो भी proteins होंगे, let us say that total 5 proteins are involved. ठीक है? So we know that when these 5 proteins are getting activated, we are getting our cell proliferation fate. Now second fate क्या हुआ ERK pathway का? To have neuronal differentiation. Now, since it is neural differentiation, obviously a cell is getting differentiated. It means it is having some destined proteins targeted, right? Uski cell surface में, and to carry out that differentiated function. तो उसके लिए वहां पे additional 15 protein और लगेंगे. This is just the rough estimation, okay? So that's why this duration is also increasing. Okay, यहाँ पे 5 ही था, यहाँ पे 15 ही था. So to activate and to make sure that these 15 proteins are also functioning properly in order to get the differentiated cell, it is taking more. So along with that, the amplification of these proteins, it is also the number of proteins involved is also under consideration. So यानी कि यहाँ पे 5 होगा तो यहाँ पे 15 होगा. ठीक है? So that's why the duration is important of any particular cell signal. Clear now?
Yeah, ma'am.
Okay, now networks and the crosstalk. So crosstalk refers to the interaction of one signaling pathway with the another, which integrates the activities of different pathways within the cell. So there is an extensive crosstalk between the PI3 kinase, AKT, mTOR, RAS, RAF, MEK, ERK pathways. Okay. So these two signaling pathways are activated downstream of tyrosine kinase receptors and play important role in cell proliferation and the survival. So crosstalk between them includes both positive as well as negative points of regulation, which serve to coordinate their activities within the cell. So what they want to say over here is that if certain proteins are getting activated, the rest other set of the proteins will be getting inactivated. ठीक है, तब ही आपका regulation हो पाएगा. Okay, so let's see over here. So PI3 kinase हमने देखा है और हमने अभी MAP kinase pathway देखा है. Now let's see how they both are interacting. So we have got the RAS, RAF, MERK, ERK pathway and PI3 kinase pathways, which is connected by the negative and as well as the positive crosstalk. ठीक है. So what do we see? Growth factor है, phosphorylation हुआ यहाँ पे आपका tyrosine kinase का autophosphorylation, PI3 kinase comes and binds. So if we remember correctly, SH2 domain of your PI3 kinase will get activated. PI3 kinase will go and PIP2 को PIP3 करेगा, and then you know वहाँ पे आपका mTOR C2 gets activated, PDK1 gets activated, which is going to activate your AKT. Now AKT, activated AKT is gonna inhibit the activity of TSC, is going to inhibit the activity of RAS. Okay, but in turn, the RAS is going to activate your PI3 kinase. Did you get it? So in this pathway, RAS is activating RAF, RAF is activating MEK, MEK is activating ERK. But here ERK, phosphorylated ERK will activate your mTOR C1, but it will inhibit the activity of TSC. So this is how when many pathways are working together, okay, when they are, you know, merged together, there is a common signaling, downstream signaling molecules are present, and that's how they regulate each other. Clear? This is just for the understanding. You don't have to, you know, remember like how the crosstalk between ERK and PI3 kinase is happening. What is the takeaway message is that when 2 or 3 signaling pathways are working together or they are connected, both positive and negative crosstalk is happening. What does it mean? If ये एक चीज आपका इसको activate कर रहा है, तो eventually उसका second step में वो उसको inhibit भी कर रहा है. ठीक है? If here this pathway is inhibiting the activity of TSC and mTOR C1, the RAS pathway is activating the activity of mTOR C1. Clear? Did you get the main concept over here, like how the negative and positive crosstalk is happening between the two pathways?
Yes, ma'am.
Yeah. So the extensive crosstalk between individual signal transduction pathway means that multiple pathways interact with one another to form signaling networks within the cell. So the full understanding of cell signaling will therefore need to go beyond the analysis of individual pathways to the development of network models that predict the dynamic behavior of the interconnected signaling pathway that ultimately result in biological response. And guys, this is not that easy to work on. Okay, so I remember in my lab, one guy was working on this only, the pathways. So he was working on only 8 pathways, which was related to diabetes that time, okay, during my postdoc. So that time I used to wonder like how to understand the crosstalk between the pathways, right? Very simple method he has developed was in the mathematical way. Okay, so what they do is that they make certain equations. Key, if suppose let's say 3 pathways are there, they give some x, y, z or something like that. If here 10 molecules are there, here 5 and here 1 is there, so x1, x2, x3 and यहाँ पे क्या हो जाएगा आपका y1, y2, y3 like that till 5 and 3 में हो जाएगा, let's say 1 is there, so only z1. Okay, now how they are connected, at which stage they are connected. So if I say x1, y2 and z1, so how is it then? X1 aapka suppose, I don't know, let's say it is PI3 kinase only. PI3 kinase is activating some, let's say AKT, and then this is, you know, activating some other protein. Okay, so यहाँ पे अगर आपका plus and minus है, so plus means activating, minus means inhibiting. So that's how they used to do, and they put into a mathematical model. Model मतलब यही equation बनाना. And when you put these equations in the Excel sheet and you try to make a graph, then some ऐसे कुछ तो graph मिलेगा. And from that they understand like, you know, how crosstalk can be done between the different pathways. Did you get it? I mean, this is what I understood. मुझे भी high level का नहीं मालूम है, but this was my understanding. If I consider only 3 pathways, so now we know that there are many multiple pathways are there, and it is not possible for you to do in the wet lab. यानी कि हर एक pathway को आप study करो inside the cell, you know, doing all the experimental activities. It's not possible. So it's better to go with the mathematical modeling. So when you say mathematical modeling for any biological concept, you actually derive an equation which satisfies the biological conditions. Okay. And whenever you put that equation, अभी जैसे कि यहाँ पे मैंने कितने डाले थे 10, 5 and 1 डाला था. Now let's say that there are 100 components, 25 components, 5 here. How this pathway is going to look like? What will be the complexity of this pathway? So that you can understand when the equations is there. So you put in the equation these numbers and then you derive the answers, like, you know, what you get. So based on that, you can understand the complexity of the signaling pathway. Clear, guys?
Yes, ma'am.
Yeah, this is really very interesting only if you do understand the, you know, the application of maths in bio. So then it becomes really very interesting to do such research. So where you don't actually have to put the real experiment, but of course you need to understand the experiment. Okay, it's really interesting. So here I'll just explain you, okay, this thing a little bit more in detail. Okay, so how do you start this thing? So let's say that we are talking about cancer only, and let's say that you're working on lung cancer. Okay, so we know that there are many cancers and lung cancer में भी बहुत तरह के होते हैं. So let's specify one type of lung cancer and let's say that you're working on EGFR gene mutation. ठीक है, so EGFR आपका कौन से signaling pathway में आता है? Okay, that you're going to identify. Let's say that there are 5 signaling pathways in which EGFR is involved. Now for that you need to do the literature search. आप research publication पढ़ोगे जिसमें से आपको पता चलेगा EGFR के कौन से signaling pathways है. Now these signaling pathways, let's say that you name it as A, B, C, D and E. Okay, now each pathway, at which stage, how many molecules were involved in this pathway? 10, किसी में 5 होगा, किसी में 25 होगा, किसी में I don't know, let's say 14, or let's say, you know, 4, I don't know. At which stage EGFR? So basically EGFR will be the your ligand. Okay, mainly तो वही होगा, but again यहाँ पे कितने आपके signals हो रहे हैं before, you know, binding to the receptor. Okay, so that can be also included. Then you give these number as A1. Whether it is activating तो ये plus हो जाएगा. If it is not activating वो negative हो जाएगा. Now permutation combination of A1 with these 10 with these 5, when this 25, 14 and 4. Okay, so all those combinations you are gonna make and you derived an equation कि at the end में क्या होना चाहिए, that if there is a mutation in the EGFR, eventually the cancer cell is surviving. Okay, the cell is surviving. So all those permutation combination you're gonna make. So इससे हमें ये पता चलेगा like how our anti EGFR drug should work. Are you able to correlate? These combinations are gonna give us the high probability of cancer cell survival. So as a research scientist, we should understand that the discovery or the synthesis of the drug should be in such a way that it is going to overcome all this possibility or the probability of cancer cell survival, so that this drug can inhibit this survival, or in short, killing the cell. Okay, so when we do any research, it is not always the you know, the experimental work which gives you the data. Basically it's the stats, it's the maths which gives you the idea कि हाँ ये drug कहाँ पे act कर सकता है और ये कितने तरह के EGFR mutation causing lung cancer में act कर सकता है. Okay, so it will tell you the combinations. Okay, let us say that, for example, let's say that there are 45 cases in which this drug is gonna act out of 1 lakh. So it's a good chance, right? So at least out of 1 lakh, you can save these many people having those combinations with this drug. And for the other rest 55, you can think about it, either change the structure of the drug or add some other functional group so that it can also overcome all these other 55 combinations. So guys, did you get it? The story over here, like how in research, the research which is the application is they try to bring into the market. So when I say market, matlab direct for the welfare of the mankind. Okay. Yeah. Krishita, did you get the idea? So like how research is gonna help eventually. So it's not
Yes, ma'am.
not like you know 6 महीने या 6 साल में नहीं है. This is like over long period of time. Okay, so that's why we publish paper. अगर suppose एक lifetime में तो पूरा काम होता नहीं है. So let's say that I worked on 2 proteins. So I understood the structure and function of the protein, and I said that these are the applications that you can use it for the further research. Okay, so जो मेरे बाद आएगा वो उसको application level में search करेगा. They will try to design experiments in order to help people. Then they can, if when they get the good answer or good results, then they can bring that particular drug into the market. Or if it needs any modification, so then it goes to the organic chemistry part where the modifications can be made, and then eventually they do the trials in the in vitro cell lines first, and then it goes into the small animals like mice, mouse or rat or hamster, and then agar wahan se bhi approve ho gaya, then it goes in the monkeys and chimpanzees, jahan hone ke baad then it enters your phase 1, phase 2, phase 3, phase 4 of your clinical trials. ठीक है. phase 4 पहुंच गए तो समझ लो almost it is accepted by the US FDA to be used for the treatment in the human patients. Okay, yeah. So it's a long procedure, and COVID-19 का drug इतने जल्दी आना, it's actually a miracle, because within a year, 2 years, it was brought into the market and people have used. Otherwise, कोई भी vaccine, कोई भी drug को market में आने में for the first time, it takes so many years, around 15 to 20. Now, why COVID-19 was easy to bring into the market? Because already, if you remember, Middle East respiratory syndrome and severe acute respiratory syndrome, SARS, MERS, already they have caused so much of death in history. So that time those structures were available. उस virus के structure available थे. And corona, this COVID-19 belongs to the same family. तो इसलिए structure easily available था. Almost those vaccine, those drugs were also available. So that's why bringing the vaccine into the market was easy. Easy in a sense मतलब अर्ली, लाइक उसके लिए हमें 15 साल वेट नहीं करना पड़ा, नहीं तो आज हमारी पॉपुलेशन और 1/4 हो चुकी होती इफ वी वर वेटिंग फॉर अनदर 15 years, right? So it's a huge advancement in science and technology. Okay, any doubts over here? Yeah, so with this topic is over, and tomorrow we'll start with the cell cycle. I would have taken more actually, but 1 second.